Southern blotting probes. Labeled material to detect a target is called as probe. In case of DNA, usually 20 to 30 nucleotides long complementary sequence to the target is used as a probe. Here we can see that what is a probe. This is the DNA fragment, or we can say this is the target DNA sequence, and this is the probe. So probe is actually complementary sequence to the target DNA. If there is C, here we can see that G. C to G, they are complementary to each other. So probe is complementary sequence to the target DNA. There are two types of the probes. Probes can be radioactive probes, like in case P32. This type of the probes, they are sensitive, they are relatively cheap, they are hazardous, and radioactive waste disposal regulation should be followed when one have to use a radioactive probe while performing the Southern blot technique. While on the other hand, non-radioactive probes are also available, like biotin. This type of the probe is sensitive, relatively expensive. Hybridization of the probes. The binding between single-stranded labeled probe to a complementary nucleotide sequence on the target DNA is called as hybridization of the probes, because the sequence of the probe and the sequence of the target, they are complementary to each other. If they are complementary to each other, then the probe will bind to the target sequence. While if the sequence of the probe is different and it is not complementary to the target, it will not bind to the target sequence. Probes are the labeled material to detect complementary sequence on the DNA.